Hello everyone, how are you? So in this lesson we shall study about the gene mutations. This course is presented by me, Navneet Sharma. You can know more about me from here. You can follow me on this link, and if you like the lesson, kindly rate, review and recommend. So beginning with the lesson. Nowadays the word mutation is related to gene mutations. If a nucleotide sequence of DNA molecule undergoes a change, it is called a mutation. Now the gene is called a mutant gene. In 1909, Morgan observed white-eyed Drosophila. This white-eyed color is a mutant character. The mutant gene and the original gene will be present at the same locus of chromosome. The mutant gene is always recessive. Only in the homozygous condition, the mutant gene will be expressed. Now the gene mutations are of two types in nature. Mutation arise by themselves. They are called as spontaneous mutations. Other than that, we have induced mutations. By the applications of mutagenic agents, mutations are brought forward. This is called as induced mutation. In 1927, Mueller brought induced mutations by X-rays in Drosophila. For this, he awarded Nobel Prize in 1928. Stadler introduced induced mutations in barley plant by X-rays, and Orbach used mustard gas to bring mutations. Many chemicals will cause mutations. Okay. Reverse mutation. A normal gene will undergo mutation and become a mutant gene. After some time, this mutant gene will undergo mutation and becomes an original gene. Now this is called as the reverse mutation. For example, in a wild pea plant, white flower is because of the original gene. Now because of mutation it becomes purple, and because of reverse mutation it becomes white. Characters of a gene mutation. Mutation occurs at random. A direction cannot be predicted. It will not occur to fulfill a specific requirement. The mutation is because of change. Usually the mutant genes are always recessive, mostly. Okay. Usually the mutations are harmful. Approximately 99% of mutations are only harmful. Okay. Mutations will form a raw material for evolution. Okay. Mutation on a regular basis in the offsprings sometimes produce evolution. DNA is the basic material of a gene. DNA is responsible for hereditary characters. In a DNA molecule, a specific base sequence is present. It will not change through many generations. Now, a DNA molecule is made by two polynucleotide chains. In each strand, 4 kinds of nucleotides are present. The genetic property of a particular genetic molecule of DNA is determined by the specific sequence of its nucleotide bases. Changes in this sequence or on the base pair is changed, mutation is caused. Now, Watson and Crick predicted that a base pair change at any point, but it is very rare. This is represented in the following way: that AT is changed into a GC pair. It is not a very easily occurring process. Okay. This type of change is called a mutation. This is carried on to a point, and hence it is called as the point mutation. Because of its invisible, it is called as invisible mutation. This is also called as the gene mutation. This is brought forward by the following methods. In DNA, the base pairs are AT and GC. Here is G. Okay. In each of the 4 base molecules, various atoms are arranged in a certain manner. A single shift in this arrangement as a result of hydrogen atom moving from one position to another position within molecule will cause the change in the form of base. This process is called as the tautomerism. The 2 forms of base are called as tautomers. Common tautomeric forms of G and T are keto compounds. The tautomeric forms of A and C are the amino compounds. Now and then enol forms amino forms may occur. These are called as abnormal tautomers. Because of these abnormal tautomers, gene mutations are brought forward. Okay. In these tautomers, the mobile hydrogen atom is changed in their position, cause of which the tautomeric form will show abnormal behavior. The normal pairing is A, T, T, A, G, C and C, G. The tautomeric pairing shows A, C and G, T, etc. Unusual pairing during replication. Now transition GC-AT pair as a result of incorporation of a rare tautomer A during replication of DNA will cause a point mutation. Now during replication of DNA, GC will separate, G combines with C, but C will combine with the rare tautomer A, At this will bring a mutation. This change is seen in the following way. Some unnatural bases are chemically related to natural bases. Their chemical structure is related, but their behavior is different. For example, thymine 5-bromo uracil substances are chemically related. They are called as analogs. Thymine combined with A, but its analog 5-bromo uracil will combine with G. In this way of the function, they are differ, but hence by using the base analogs, we can bring mutations. A and I pair can be changed into GC pair, AT pair can be changed into GC pair by using by using a 5-bromo uracil. When bacteria are grown on 5-bromo uracil medium, mutations have come. Bromo uracil are incorporated in place of T, because of which in the replication AT pair changes into the GC pair. Now some chemical substances will bring mutations. They are called as nitrous acid hydroxylamine. When a bacterium is treated with a nitrous acid A, it will change into a GC pair. Nitrous acid will act on the adenine and convert it into the xanthine. The xanthine will combine with cytosine. It brings point mutation. During replication of DNA molecule, GC pair will produce now and then A may combine with G, which is very rare. In the next replication, then is corrected. This simple mistake in first pairing will convert AT pair into the GC pair. There is a lot of typing mistakes, sorry for that. Two bases of a pair will break their bond with sugar. When they unite with sugar, they will show 180 degrees rotation. This causes CC pair become GC pair, or AT pair will become a TA. Okay, this is called the transversion. Okay, here is okay, GC pair will become a CG pair. Okay, in this way point mutations will occur in an organism. So this was a rather complicated process. So if you revise it again, you will understand it simply. So I hope you guys like the lesson. Keep learning.